Does anyone know how to make a slide to observe microorganisms in water? First, we'll need a glass slide, a cover slip, a dropper, and tweezers. Use the dropper to take a small sample of water from a pond or a potted plant. Drop the water onto the glass slide, and then, using the tweezers, gently place the cover slip on top. Finally, use a tissue to absorb any excess water around the edges. And just like that, you've made a slide to observe water microorganisms. Now, please follow these steps to make your own slide for observing microorganisms in water. Under the microscope, we can see some impurities and algae. Everyone, look over here, there's a paramecium. The name paramecium comes from their oval or slipper shape, and they move using tiny hair-like structures called cilia. The paramecium's body is very small and covered by a membrane. It's a typical single-celled organism, made up of just one cell. If you observe closely, can you see that the paramecium's body has many unclear structures? To help you see them more clearly, I've drawn a diagram of its structure. Look, on the paramecium's membrane, there are cilia, and inside, you can find the micronucleus, macronucleus, contractile vacuole, collecting canals, cytoproct, food vacuole, and oral groove. Do you know what these structures do? The paramecium uses the movement of its cilia to spin and move forward in water. The membrane helps it take in oxygen and release carbon dioxide. The oral groove is where it takes in food. The micronucleus contains genetic material, while the macronucleus controls the intake and distribution of nutrients. The contractile vacuole and collecting canals remove waste and excess water. The food vacuole helps digest food, and the cytoproct expels undigested residues. Each structure in the paramecium has its own job, enabling it to survive well in water. Besides Paramecia, many other microorganisms are in the water waiting for us to discover. Next, you can exchange the slides you made with your classmates or use different water samples to create new slides and observe even more microorganisms.